Bismillahirrahmanirrahim. Rabbi shrah li sadri wa yassir li amri wahlul uqdatam min lisani yafqahu qawli. Assalamu alaikum students. This is the first lecture of Bio 310 Introduction to Bioinformatics course. In this lecture we will discuss about what bioinformatics is. We'll discuss a short history about how bioinformatics field emerged, what are its aim, what are its applications in the field of biology. So let's start. You will find different definitions of bioinformatics on different websites and even in books. We will discuss some of those definitions here today. One of the definitions says that bioinformatics is an interdisciplinary field that develops methods and software tools for understanding biological data. So this is one aspect of bioinformatics where the people, they develop different methods, they write different algorithms, they develop certain tools and softwares in order to understand the biological data which is being produced in exponential numbers now these days. So this is one of the aspects of bioinformatics. Another definition of bioinformatics is simply the union of biology and informatics, which means that bioinformatics involves the technology that uses computers for storage, retrieval, manipulation, and distribution of information related to biological macromolecules such as DNA, RNA, and proteins. In the subsequent lectures, we will discuss why there is a need to store the information, why storage is necessary, and then how to distribute that information to the people around the world, and how we can retrieve the data. We'll have some theoretical lectures, and also we'll do some practical work in the lab. So we'll discuss these aspects in subsequent lectures. Another definition which is very precise but comprehensive, which I really like, is that bioinformatics can be considered as a field of data science for solving problems in biology and medicine. So the data science is vast field, and its application is very vast. They can be used in computer science, they can be used data science can be implemented in our daily life. Similarly, the data science can be also be used to study biology and medicine and to solve complex problems within it. Uh, Another definition says that bioinformatics is an interdisciplinary field of science where bioinformatics combines computer science, statistics, and mathematics to analyze and interpret biological data. So if we focus on the last definition that we have just discussed, so if as you can see in the Venn diagram, so there are three separate fields. There is statistics, computer sciences, and biology. So when stats and computer sciences, they merge together, or when they overlap, a new field emerges out of it, which is known as data science. Similarly, computer science and biology, when they are merged together, the field that pops up is the computational biology. Similarly, biology and statistics, when combined together, it becomes biostatistics. But when biology, statistics, and computer science, it's they all three fields, when they are brought together and merged with each other, then a new field of bioinformatics emerges from it. Okay, so bioinformatics is basically a combination of or integration of biology and study of complex biological processes using computer sciences and statistics as tool. Okay, and this is what generally bioinformaticians do. This is one of the aspects that the in this figure on the right side, you can see that there is a whiteboard, and there's a flow chart like sample preparation and sequencing and raw data, and then gene identification and whole gene discoveries. So this is you can see that standard workflow that is being followed these days. And from the job of bioinformatician is to extract meaningful information from the raw data. Okay, so we have tons of data present on the internet, and we also have we generate tons of data every day. But generating data is one thing. extracting or mining meaningful information out of that raw data is what is the job of bioinformaticians. And using these computational and statistical tools, bioinformatics people or bioinformaticians, they take up the raw data, analyze it, and on the basis of that analysis we are then able to identify certain new genes or novel gene discoveries, and we can also um, study different biological processes. uh, so let's talk about bioinformatics and its brief history. So the term bioinformatics was originally coined by Pauline Hodgbeck and Ben Hesper, and they started to use this term of bioinformatics in 1970s. And by bioinformatics, what they meant was that it is the study of informatic processes in biotic system. Okay, so we all know that you have studied the introduction to molecular biology course, and you know that about the central dogma that information flows from DNA to RNA and then RNA to proteins, and then we can have also um, reverse transcription process, and we have replication process. Okay, so there's a flow of information in biotic systems, and to study that those processes, um, using computational tools is what the scientists they use the term bioinformatics for this kind of study. But even before coining the term bioinformatics in 1970s, uh, the field of bioinformatics was there at least a decade before that, because people they were started to use computers as an aid to study biology, but there was no proper term as bioinformatics at that time. Okay. So there were three important factors that facilitated the emergence of computational biology during early 1960s. Okay, so these are the three main factors that facilitated the emergence of this field. The first one is that the idea that macromolecules carry information. Okay, so initially people, when DNA was not known to be the main hereditary material, but people scientists they had the idea that the macromolecules such as DNA or RNA or proteins, they carry the information, and this information is being carried from one generation to another, and so on and so forth. And the second point that facilitated this field was the expanding collection of amino acid sequences. Now you must be wondering why amino acid sequences. Earlier, when it was not proven that DNA is a hereditary material, scientists were more inclined towards in believing that proteins they are basically the main information molecule or the hereditary material, because a DNA was so-called boring molecule. It has just four nucleotides: adenine, guanine, cytosine, and thymine, and it was just a polymer, a long polymer with repeating, nucleotides. Whereas in proteins, when they studied proteins, they saw that there are 20 different types of amino acids, and amino acids have different properties, and they can be, placed with each other in different combinations, and the resulting proteins are versatile in nature. um, they are complex, they have different functions to perform. Okay, so these properties of proteins and versatility of proteins led scientists to believe at that time that proteins are the hereditary material and they are the main information molecule. But not all scientists believed in that. There were some scientists that that believed that no, the DNA is the hereditary molecule, and they both these groups they were working in that direction. And the third main factor that facilitated in the emergence of computational biology was the development of computers, high-speed digital computers. Though they were not high speed if we compare the computational speed which we have today, but there were computers at that time, and computers were getting better day by day, right? So these three are the main points that facilitated the emergence of this field in 1960s. And let's refresh our memories, and um, you must you have you will recall that in your introduction to genetics course and molecular biology course also, that there was a very uh, brilliant experiment by Avery, McLeod, and McCarthy in 1944, where they used different strains of bacteria, pneumococcus bacteria. So there were two types of bacteria. One were the round shaped, or one were the smooth, and the other one was rough. So rough strains were non-virulent and smooth strains were virulent. So what they did, they injected a rough strain or non-virulent strain in mice, and they saw that the mice lived. And when they injected the smooth virulent strain into the mice, the mouse died. Then what they did, they killed the smooth strain by heating it. So heat killed smooth strain when they were injected into the mouse. So as per their what they um, expected, they expected that the mouse will live, and actually the mouse lived. But then there was another combination where they mixed a rough strain which was non-virulent with a smooth strain which was heat killed. So both became non-virulent. So rough strain non-virulent plus heat killed smooth strain which is also non-virulent. When they injected this mixture into the mouse, they expected the mouse to live, but what they found out, or they got the result, that the mouse died. So they thought that there must be something transforming into the mouse, and and from there they presented a transforming principle, that there was something that changed non-virulent strain into a virulent strain. And obviously then subsequent experiments, they uh, did some more experiments and found out that when DNA was removed, or damaged, so that then the mouse lived. You can go and Google this experiment if you want to study more about this experiment, to learn more about this experiment. Then in 1952, two scientists, Hershey and Chase, they did a brilliant experiment where they used bacteriophages, and there were two types of bacteriophages. One had the protein code labeled, and the other group had, the genetic material or DNA within it was labeled. So the DNA was phosphorus labeled, and the protein code was sulfur labeled, and they infected the bacteria, and they found out that uh, it was the DNA that was the genetic material, not the proteins, because proteins were left out, and it was basically the DNA that used to go into the bacterial cell and multiplied, and you know ruptured the bacteria. So this showed that DNA is actually the hereditary material, not the proteins. And then in 1953, the Francis Crick and James Watson, they presented the double helical structure of DNA, and from then it was known that no, it is the DNA that is the hereditary molecule, and it has all the information in it. Uh, but as discussed earlier, that protein analysis was the starting point, because at that time protein was very a protein still is a versatile molecule, and at that time it was um, believed that protein is the hereditary molecule. So a lot of work was being done on proteins. So first sequence of protein, that was insulin, it was published in 1950s. And atlas of protein sequence and structure, uh, that was developed in 1965. And then in 1970s there was a development of a protein data bank. So this was developed in Brookhaven National Laboratory, and initially it contained only a few dozen protein structures, but now the number of protein structure in PDB is more than 30,000. Okay, so there is an exponential increase in the number of protein structures in PDB. Then in 1970s, there was a first sequence alignment algorithm that was developed. We will discuss why sequence alignment algorithms are useful, and how they are useful, and how do they actually work. So first sequence alignment algorithm was developed by that is Smith Waterman and Needleman's Wunsch algorithm, that they were developed in 1970s. And first protein structure prediction algorithm was developed in 1974 by Chow and Fassman. Okay, so this is a brief timeline. I have not added many things, many time points in our history part. These are just a few time milestones, um, which are important and we should know. Among many scientists that working at that time, Margaret Diehoff was can be considered as the first bioinformatician, and David Lippman, who was a director of National Center of Biotechnology Information, he called her both father and mother of bioinformatics because of her enormous contributions in the field of bioinformatics. Some of the major contributions are listed here, which is number one is she is the pioneer of application of computational methods in the field of biochemistry. So she was the first person who uh, um, used computational tools in the field of biochemistry. And second major contribution of Margaret Diehoff is her collaboration with Robert Daly, who was a physicist, and it resulted in the development of a computer software known as COMProtein, and it is a computer-based program designed to determine the protein primary structure using Edman degradation method of peptide sequencing. So I'll not go into the details of what is Edman degradation. I hope you might have studied this uh, method of sequencing proteins. This is a chemical method. So briefly, a protein is cleaved or is broken down into smaller fragments, and then those fragments are sequenced. And after sequencing of those different fragments, these fragments are you know they are aligned with each other to see what actually was the sequence of the protein. So the COM protein was the first computer program that was designed to determine the protein primary structure. So they used to enter the fragments of DNA, proteins, sequence of amino acid into the software, and the software used to give you output of the primary structure of the protein, which is the amino acid sequence of that protein. The third major contribution of Margaret Diehoff was the development of a one-letter amino acid code that is still in use today. So, if a three-lettered code was used, in COM protein, so it made the computational process complex. So, in order to reduce the complexity and make the algorithm work much efficiently, she introduced a one lettered amino acid code, which made the software much more efficient, and it was then easy to use. And she was the one who published the first atlas of protein sequence and structure in 1965. So these are just a few of the contributions Margaret Diehoff had in the field of bioinformatics, um, among many other contributions. So this is the picture of ComProtein bioinformatics software. In the figure panel A, you can see large computers, and in the figure panel B, they were there are different punch cards. Okay, so it's an old system, old method of um, how computers used to work. And in the figure panel D, you can see that there are four different fragments of amino acid sequences. So in the fragment 1, there's threonine, histidine, glutamine, and cysteine. And this fragment number 2, there's a glutamine and cysteine and alanine and threonine, and then we have lysine, threonine, histidine, and then we have methionine, isoleucine, and histidine. And when they were rearranged, and when they are placed in proper way by uh, using the overlaps, you can see that the uh, primary structure of protein turned out to be methionine, isoleucine, lysine, threonine, histidine, glutamine, cysteine, alanine, and threonine, which is shown in the red. So this software used to take input of as a different short amino acid sequences, and it turned out, or it gave the output as the whole primary structure of the primary sequence of the protein. So we are in the age of in silico biology. Biology is becoming, a quantitative science now, and it has transformed it from observational science to experimental and then to data science. Now, to understand this, this point, we can take the example of Mendel's law. So, in 1800s, you've studied Mendel's law of inheritance in your genetics course. So what did Mendel do? He planted many pea plants, and it was basically his observation and mathematics. So he observed, and doing statistics and mathematics, applying mathematics and statistics, he came to know that okay, there's a 3:1 or 9 is to whatever is the in the law of segregation, and it gave a law of independent assortment, and there was a pattern of inheritance. So that was pure observation, and then scientists, based on these observations, they did experiments to prove that those observations were correct, and the inheritance material was later turned out to be the DNA, and then we knew that okay, there are genes, and genes are placed in specific order, and we know that there is a process of mitosis and meiosis and everything, right? So from observation, it was transformed into experimental science, and now experimental science is changed or transformed into a data science. So these days we have a lot of information coming out, and because of these experiments obviously, and a huge amount of data is being produced these days, and that data is now needs to be evaluated, it needs to be worked on to see what important information we can find from the raw data, which we have discussed initially. And new discoveries, they require large data sets and advanced analytical methods. Okay, so you need a large amount of data. Just on the base of small data set, you cannot jump to conclusion. Okay, so in order to jump to a conclusion, you need to have a large data set, and based on those large data sets and analytical methods, then you can get to new discoveries. Okay, so bioinformatics is now a multidisciplinary field, and it has become very important, and you can you can with the help of this figure, you can understand that the genomic data has increased exponentially, and this is the power of genomics. And because of this increase in the genomic data, it has also created many jobs in the last decade. And sequencing is it's continuing, it has not been stopped ever since the method of sequences was developed, and it's generating tons of data every day. So the figure that is shown on the left side, the graph on the left side, you can see that when the Human Genome Project was started, so the cost of genome sequences was very high. So with the passage of time, the cost of genome sequences, was getting lower and lower, and now you can see that it has come to $1,000 per genome on average. Okay, so cost of genome is has reduced, and similarly the genomic sequences, genomes that have been sequences since Human Genome Project, they have um, you can see an exponential growth there. Okay, so because of low cost, there's a high output of genome sequencing results. Okay, so this is the power of genomics, and this is you can get an idea from this figure that why the field of bioinformatics is important nowadays. Okay, so obviously this bioinformatics It's a combination of biology and information science, and the overlap is getting more day by day. Okay, so with the passage of time, the merger of biological science and information science is getting higher day by day. It's getting deeper day by day. So what are the goals of bioinformatics? So the ultimate goal of bioinformatics is to better understand a living cell and how it functions at the molecular level. Okay, so with the help of bioinformatics, uh, this point will get more clear when in our subsequent lectures, when we will discuss different aspects of bioinformatics, when we'll discuss different algorithms and tools, and when we'll apply those tools in our practical sessions, we'll see that the bioinformatics, the goal of bioinformatics is to to better understand a living cell and how it functions at the molecular level. So using different tools and methods and algorithms, we can achieve this goal. And the scope of bioinformatics can be divided into you know two different directions, and they can be two different directions or scope. One is the development of computational tools and databases. So this is majorly done by people who are good at computational side, who have interest in computers, and their statistics and mathematics is strong. So these guys they are more into development of computational tools and databases. Okay, and then there's another direction which is taken by the biologist like us, that we are in at the user end. We are we are at the application of these tools and databases. So what we do, we uh, use these tools in our research. Okay, so we use the tools, we use the databases that are available online, and apply these tools and databases to our research to get the results that we um, want to have. Okay, so this is the scope of bioinformatics, and these are the applications. So broadly speaking, there are application of bioinformatics can be divided into three major components. One is the structural analysis, the second one is the sequence analysis, and then we have the function analysis. So structure analysis, we have nucleic acid structure prediction, we have protein structure prediction, we have protein structure classification, and then protein structure comparisons. Okay, so in this direction or this line, we are more interested in the structure of macromolecules. And why the structure of macromolecules are important? Because structure has it has influence on the function of the protein. Then we have sequence analysis, where we sequence the genomes and we compare different genomes. We study the evolution of those genomes and sequences, and we construct phylogenetic trees to study okay, how these sequences they evolved in the history. And then they have gene and promoter prediction and motif discovery and all sort of uh, analysis which are related to sequence. Okay, and the third main application of bioinformatics is the function analysis. Okay, so we have the sequence analysis, we have the structure analysis. Now we have to predict, or we have to study the function of those molecules, because studying the structure and sequence is not enough. It's not it's incomplete job. So now we know the if we know the structure of a protein, for example, and we know the sequence of the protein. Now the next step is to study the function of that protein, which is very important. So in function analysis, we have metabolic pathway modeling, we have gene expression profiling, protein interaction predictions, how proteins they interact with each other, and these interactions, they are very important for functioning of the proteins, and how proteins and where proteins they have they are localized in within the cell, and we predict all this kind of stuff using bioinformatics, right? So these are basically the application of bioinformatics. Bioinformatics as a field is cool. Okay, so it offers flexibility and you can work from most places. You don't have to do much science. So what do we mean by these statements, that all you have to all you require is a good workstation, a good computer with good specs, and you can work from your office, you can work from your home, and even you can work while sitting in a coffee shop. Okay, so you can do bioinformatics. Unlike people who are from the wet lab side, they have to be in the lab and they have to run the experiments. So bioinformaticians, they don't have to do much science. They don't have to run experiments and um, uh, follow all the protocols and do the preparation and do experiments and this stuff. So the experiments are being done by the people in the wet lab, and the people and the bioinformaticians, they use that data and extract the information and give back the meaningful information to the um, people in who work in the wet lab, and then they do further experiments to find out what's there. Okay, so since you guys um, are from the biosciences background, and you'll be doing some wet lab, so you don't have to lose heart. Sometimes you get some opportunity once in someone's lifetime, I would say, when bioinformaticians, they will be working hard, they will be working more, and the biosciences people and the wet lab people, they will be enjoying their days at home. So this was just in lighter mood to end this presentation. I hope you have learned something new. You have now the idea what bioinformatics is, what is this field, and what are its applications and scope. And I hope you enjoyed this lecture. If you have any questions, write it down. We will discuss your questions, and if you have any queries, we'll discuss and address to those queries in our QA session next week. Until then, Allah Hafiz and enjoy your time.